wait for a minute. Yeah, we are live. You can start, Anshika.
Okay. Good morning. Welcome to the 2-week online faculty development program, Chemistry, the Catalyst for Change. I would like to welcome Dr. Bijay Lakshmi Nanda, Principal, Miranda House, and Dr. T.K. Mishra, Vice Principal, Ramanujan College. Thank you so much, ma'am, and thank you very much, sir. I would also like to welcome Dr. Anant Narayan Bhatt, Scientist F at the Institute of Nuclear Medicine and Allied Sciences in Mars, Defense Research and Development Organization, DRDO, Delhi. He is our first keynote speaker after the inaugural session. Thank you so much, sir. The Teaching Learning Center, Ramanujan College, in collaboration with Department of Chemistry, Miranda House, is organizing a 2-week online faculty development program, Chemistry, the Catalyst for Change, under the aegis of Ministry of Education, Pandit Madan Mohan Malviya, National Mission on Teachers and Teaching. One of the main objectives of this mission is to facilitate teacher training by constituting teacher learning centers in various institutions of higher education in our country. The teaching learning centers, TLCs, are mandated to promote on a continuous basis the learning of new pedagogical practices, methodology of forming discipline-specific curricula, and creating new learning material which benefits faculty members teaching in colleges and postgraduate departments. To promote the inclusive nature of higher education, the TLCs will aid faculty in capacity building for curriculum design, scientific assessment, evaluation, and development of new academic programs. Ramanujan College, a premier institution of the University of Delhi, is located in the well-known area of Kalkaji in South Delhi. The college runs 16 courses in different subjects in humanities, commerce, and science streams. It is also the study center for the students of Non-Collegiate Women Education Board, University of Delhi, and the Indira Gandhi National Open University. The college also runs several professional courses. The teachers of Ramanujan College are highly motivated and have original, published, academic and creative work, including journals and other print media articles and educational film making to their credit. Ramanujan College has been accredited Grade A by the National Assessment and Accreditation Council, NAAC. Apart from ensuring academic excellence, the college emphasizes the holistic development of personality, meaningful exposure to the real world, and inculcates practice skills among the students. Miranda House, College for Women at University of Delhi, is amongst the premier institutions of the country. It has been awarded All India Rank 1 amongst colleges under the National Institutional Ranking Framework (NIRF) for 4 consecutive years in 2017, 2018, 2019, and 2020. Department of Biotechnology, Government of India has given it the Star College status. The National Assessment and Accreditation Council, NAAC, has accredited the college with Grade A+. Established at the dawn of independence on 7th March 1948 by the Vice Chancellor of University of Delhi, Sir Maurice Boyer, Miranda House chronicles the history of women's education in India. The college has a rich legacy. Being a college established and maintained by university, Miranda House has a special place among the women's colleges of the University of Delhi. Its location in the heart of university campus and its close interaction with the various departments of the university and other campus colleges give it a unique advantage. It welcomed interaction with other colleges and educational organizations across the country, actively engaging both men and women in all its extramural activities, competing with the best on equal terms. The Department of Chemistry, Miranda House, was established in 1948. It currently has 3 modern and well-equipped chemistry laboratories. It also has an instrumentation laboratory with various instruments enabling the students to carry out all experiments, including in the news labels as well as some simple projects. Some of the faculty members are currently involved in research. They have been recognized as PhD supervisors and have also presented papers in international conferences. In keeping with the tradition of the department, the non-academic staff is hardworking, helpful, and committed. Chemical science is a central science and is required for practically all scientific research. Chemical science is concerned with the understanding and practice of changing one substance into another. Chemists enjoy discovering both naturally occurring and man-made chemical substances with the goal of improving human life. The subject's transdisciplinary nature makes it more fascinating and demanding for researchers all over the world. To keep up with the latest trends and breakthroughs in the field, practical, theoretical, and application-based teaching learning techniques are required. Due to the introduction of different state-of-the-art facilities, instrumentation, and new software, chemical sciences and technology have progressed tremendously in this century. Chemistry and related sciences have made chemical sciences and technology have progressed tremendously in this century. Chemistry and related sciences have made rapid progress in terms of knowledge and invention. This course will touch on few of the thirst areas. There is a rising need to provide chemistry faculty members from various colleges, universities, academic institutions with updated information and knowledge in order to develop creative ideas, enhance skills, and to provide excellent education. We would now request Dr. T.K. Mishra, Vice Principal, Ramanujan College, to address the session.
Well, thank you, Anshika. Honorable Vice Chancellor, Professor PC Joshi, Miranda House Principal, Dr. Vijay Lakshmi Nanda, Program Director, Dr. Amrita Tripathi Sheikh, eminent scientist, Dr. Anant Narayan Bhatt, convener of the FDP, Dr. Ashish Shukla, and dear distinguished participants of this faculty development program. आप सभी का हार्दिक अभिनंदन, आभार, स्वागत. It gives me immense pleasure to welcome you all on behalf of Ramanujan College. We are extremely delighted and grateful to our Vice Chancellor for his gracious presence as the chief guest of the inaugural function of 2-week FDP, jointly organized by our college and Miranda House. The topic of the FDP, the chemistry, the catalyst for change, is most appropriate and timely. Well, at this juncture, it is my proud privilege to tell you that during lockdown period, under the dynamic leadership of our principal, Dr. S.P. Agrawal, the college has performed more than the normal times. And through online, we have been able to educate and train more than 1 lakh teachers across the country. And we have a distinct way of doing things. We present knowledge in the form of prasad. And the result is that the prasad is being distributed at the nook and corner of the country, and it is well taken also. Well, even though I am not competent to talk on the subject chemistry, as I never studied chemistry, I think that it has a key role to play in understanding everything about vaccine or drug discovery and development. Chemistry is driven by the knowledge of molecules and their association with the life process. Drug discovery has undergone many changes over the years, but the goal has remained the same: to uncover safer medicines for improving the human life. Similarly, many institutes have recently sprung up, have come up to study nanoscience, nanotechnology, and all having a strong contingent of chemists. A variety of careers similarly are available in basic vaccine research and development, clinical trials, production, and distribution of vaccine to the public. Well, the central aim of this FDP should be to continue to make chemistry more visible and representative. I once again welcome the Vice Chancellor. Maybe he joins us very soon. And all the dignitaries who are taking part in this FDP. And I wish all the success, all the best to this faculty development program. Thank you.
Thank you so much, sir, for your motivational words. I now request Dr. Bijay Lakshmi Nanda, Principal, Miranda House, to address the participants.
Thank you, Anshika. On behalf of Miranda House and the Collaborative Association with Ramanujan College, with the Teaching Learning Center of Ramanujan College, and also on behalf of the Department of Chemistry, which has brought in this unique collaboration, I would like to welcome everyone. I would also like to welcome the Honorable Vice Chancellor, Professor PC Joshi, who will be joining us shortly. And I would like to thank the Ramanujan College Principal, Professor Agarwal, and also the Vice Principal, Dr. T.K. Mishra, for letting us join together to learn together. I think chemistry is a catalyst for change. We have already said that, but chemistry department in our college has become a catalyst for change by bringing together the two departments. So I would like to thank Dr. Ashish Shukla and Dr. Anchikal for bringing the two departments together and doing this under the aegis of the federal structure of Delhi University. I think we need to learn to collaborate more and more, because that is the way in which we can grow together, evolve together, and be able to bring out the best from the university. As a DU constituent college, we consider ourselves as a part of the federal structure of Delhi University, and this is the best way to learn for all of us, including the participants who are here. I must say that I would like to then, in the order of the day, thank Dr. Amrita Sheikh, who will be the program director from Miranda House for this particular program, the teacher in charge, Dr. Sarda Sonkar, who will also be engaging with all of you here, and all the other faculty members who have joined us. But having said that, I think it will be in the order of the day to welcome our keynote speaker. Apart from the chief guest, Professor Joshi has joined here. Thank you, sir, for joining us. I would like to welcome you on behalf of the two colleges. Professor Mishra has already welcomed you, and I would like to extend my welcome to you So we are taking forward your mission of learning together. So learning together as two colleges of University of Delhi, that has been our motto. So we are learning together. Again, I would like to welcome the keynote speaker for the day also, Dr. Anant Narayan Bhatt, who is from the Institute of Nuclear Medicine and Allied Sciences from DRDO. And I will not stand between you and the speakers, but as it is said, chemistry is a catalyst for change. And I think it's also in the order of the day to say that the Nobel Prize for Chemistry was won by two women, Emmanuel Carpentier and Jennifer A. Doudna. And they have won for the gene technology, which has to do with the CRISPR method, which is looking towards improving or impacting life sciences and contributing to new cancer therapies. And therefore, I think it is significant, like Dr. Mishra has already said, it's significant. It is a learning, all of us are learning chemistry today in the day of the pandemic. There is no way we can escape chemistry. So this is also a contribution to the national education policy, which will come in where we have to learn interdisciplinary. Interdisciplinarity has to become the major kind of focus for us. Yesterday, while having a conversation with Dr. Amrita Sheikh, I said, can others join this or should it only be Department of Chemistry? She says, no, this is a learning that is specifically for the Department of Chemistry. But I think this is where that conversation will be reopened, and Honorable Vice Chancellor will guide us. How can we make this conversation more interdisciplinary or multidisciplinary, that all of us can learn chemistry through various interventions? Thank you so much, sir, for joining us, and back to Dr. Anshika to conduct the proceedings. Namaskar.
Sir, on behalf of Ramanujan College, we again welcome you, and we are indeed delighted to see you. And you value all these things, FDP, etc. This is really enlightening for all of us. Thank you, sir.
Thank you so much, ma'am, for the inspiring words.
Professor Joshi is here. You can start the introduction. Yes.
Yeah.
We are honored to have with us Professor PC Joshi, Vice Chancellor, University of Delhi. Sir, we are delighted to welcome you to the inaugural session. Thank you so much, sir. Now, I would request Dr. Ashish Shukla, Program Convener, to welcome our chief guest, Professor PC Joshi, Vice Chancellor, University of Delhi.
Thank you, Dr. Anshika. We welcome Professor PC Joshi, sir, to the inaugural session of this two-week faculty development program. Before I invite Professor Joshi to address our participant, I would like to share with you brief about his accomplishments. Professor Joshi is currently acting Vice Chancellor, University of Delhi. Professor Joshi was Professor of Social Anthropology and Head of Department of Anthropology, University of Delhi, prior to become the 23rd Pro Vice Chancellor, University of Delhi. Professor Joshi is the recipient of numerous awards and distinctions, including the Indra Priyadarshini Braksh Mitra National Award in 1987 and the Inter-University Center Association Award, Humanities and Social Science from 1996 to 1999. He has received the Certificate of Honor at the first France-Indian meet on the psychoanalysis and psychotherapy in 2007. In 2008, Professor Joshi received the Rank of Appreciation from the Department of Sociology and Anthropology, Xavier University, Antonio D. Caigan, D. Oro City, Philippines, and the Certificate of Appreciation for his research on the disaster impacts in Asia and Europe from the Faculty of Public Health, University of Indonesia in 2009. Professor Joshi has served on the UGC SHAP Advisory Committee for SB University, Tirupati, and Karnataka University, Dharwad. Recently, he was nominated as a member of Task Force, Department of Biotechnology, Government of India. Professor Joshi was the first president of the Society of Indian Medical Anthropology, Mysore, and is an executive member of Anthropologic and Folk Culture Society, Lucknow. He is a life member of the research and scientific publication in the medical and public health with a focus on anthropology of disaster development and mountain communities. He has worked in interdisciplinary areas with the psychiatrists, psychologists, epidemiologists, and co-authored papers in the area of medical anthropology, traditional medicines, impact of disasters, lifestyle disease, and antibiotic resistance, to the name a few. His research paper has appeared in the several journals of international repute. He has also authored several books. Professor Joshi has been the co-principal investigator in a WHO-funded project. He also has the distinction of discovering a Paleolithic site in Delhi, 1983. Once again, thank you so much for accepting our invitation, sir. Over to you, sir. I will invite you to address the gathering. Thank you so much, sir.
Well, I would like to start by saying, am I audible? I hope
Yes,
I'm
sir.
audible.
Yes, sir.
Yeah.
Yes, you are audible.
I would like to start by saying that you can either be living in isolation or become spiritual. And by spirituality, I mean that when you decide not to live in isolation, when you decide that you are not alone, but your existence depends upon the existence of others. And I think what great example of this could be, then we are seeing today the relation between the topmost college, Miranda House, and emerging college, which of course has a distinction of, I think, domesticating the technology of online education, Ramanujam College. So coming together of Ramanujam College and Miranda House College is a historical event. And they are talking about chemistry, the catalyst for change. Let me tell you, I also had chemistry when I was doing my graduation, but of course it was a subsidiary. And I was day before yesterday, I was in Hansraj College, where I had my chemistry lessons taught by great P.L. Soni, who had, those who are in chemistry, at least those who have done chemistry in the schools, would know about the textbook written by P.L. Soni, and he taught us. So chemistry is actually the source of life. And chemistry, of course, the whole world, is also a chemical form, in one sense. The whole world is also a chemical form. And chemistry plays a very, very important role in our day-to-day life. Whether it is organic chemistry, inorganic chemistry, or physical chemistry, these were the chemistries in our time. But now we have got many other chemistries have come up. And one of the chemistry that is very much, I think, chemistry of the day is called green chemistry, which actually is trying to give us solutions for mitigating the adverse effect of climate change. So we are at the crossroad where chemistry is going to play a very, very important role. And this particular faculty development program, therefore, not only should be limited to the students or the teachers of chemistry, but should also be, I think, rightly said by somebody, that it should also be, enjoyed by others, those who would definitely get benefit out of it. So I'm not a subject expert, but what I'm excited about, what is exciting in the whole exercise is that I think, again, the spiritualism that I was talking about, that our colleges are coming together. And they are not only they are coming together, they are coming together with the university. And I think coming together the colleges, great colleges, we say that 1 + 1 is equal to 2, but when we collaborate, 1 + 1 always becomes 11. So if two colleges are coming together, they are not going to, result into 2. Miranda House plus Ramanujam College would result into 11. And if 3 colleges come together, it becomes 111. That is the kind of impact that we are going to have. We have 91 colleges. And I don't know, you can count how much we become if 91 colleges come together. It is my dream, it is my endeavor that all these 91 colleges come together and work together and then show to the world what they are. Individually also, they are great colleges. Individually, Miranda House is second to none in Asia, in the world. But if they come together, and I think this is the process that you are starting, this is the great process that Ramanujam College particularly, I know, has been collaborating with different colleges. And this is how I think about education, it is said, there is a difference between, the money and education. The big difference is you spend money, you lose it. You spend education or knowledge, you gain it. So Ramanujam College has learned that lesson, and I really would like to congratulate, on one hand, Vijayalakshmi Nanda ji, Professor Vijayalakshmi Nanda, the great dynamic principal of Miranda House, and also Professor S.P. Agarwal, another very dynamic principal of our DU fraternity. They are coming together. They are doing something on chemistry, the catalyst for change. This is a very, I think, apt topic for these 2 weeks faculty development program. You have my full blessings and any support that you require from the University of Delhi. We are there rock solid behind both these colleges. Thank you very much for inviting me. Namaskar to everyone.
Thank you so much, sir, for your motivational words. Now I would request Dr. T.K. Mishra, Vice Principal, Ramanujan College, and Dr. Vijay Lakshmi Nanda, Principal, Miranda House, to thank the Vice Chancellor.
We have...
Well, so nice to hear our Vice Chancellor, and especially saying that these two dynamic principles, I have full backing, full support. These kinds of words, these words are really so motivational. And I think in all sincerity, he means what he says. And this is the kind of prashad that we have got with his inaugural address, and this is highly inspirational. I thank him once again for coming and sparing his so valuable time to this FDP, and whatever you have indicated, we would certainly like to follow them, sir. This is our assurance. Once again, thank you, sir.
We are grateful for your kind words, and I think this particular program or any kind of collaborative program from Miranda House owes it to your inspiring leadership. I think in Delhi University, in the present times, the kind of accessibility you have shown us, the kind of blessings you provide us is in the true tradition of Indian education, because we have learned from you and we are inspired by you to join hands. So this is a beginning. We have already had 2 faculty development programs in spite of not having a teaching learning center, Pandit Madan Mohan Malviya National Mission on Teachers and Teaching. And with Hansraj we joined, and now with Ramanujan we have joined. So we, being a DU constituent college, consider ourselves a part of the Delhi University federal structure as being one of the main colleges, and we feel that we get the special treatment because of that. So we are indeed grateful to you. I think our participants will take your words with them, and in the light of your words, we will carry forward the mission of learning together. Thank you very much, sir.
Thank you very much to all of you, and all the best for this exciting faculty development program. I wish you all the best. Thank you. Have a good day.
Sir, it is open for all the disciplines. It is not only for the chemistry. We are open for all these disciplines.
That is very good. Yeah. That is another, feather in your cap.
Thank you so much, sir.
So we should let sir know how many participants are there at present, because he cannot see that. Yes.
More than 450. More than 450 participants.
Very good.
So they have joined us from the
YouTube channel.
Very good. Very good. Thank you. Bye-bye. All the best.
Thank you so much.
So I would now request Dr. Amrita Tripathi Sheikh, Program Director of the course, to give vote of thanks. The Zoom is yours, ma'am.
Thank you so much, Anshika. On behalf of the Department of Chemistry, Miranda House, I take this opportunity to thank Professor PC Joshi, Vice Chancellor, University of Delhi, for taking time out from his very busy schedule and addressing us at the inaugural session of this 2-week faculty development program. The topic, the title, I think, Catalyst for Change. Chemistry in the sciences, and I'm sure many humanities subjects are also catalysts for change. I would like to specially thank Professor S.P. Agarwal, Principal, Ramanujan College, and Director of the Teaching Learning Center under the Pandit Madan Mohan Malviya National Mission on Teachers and Training at Ramanujan College, for giving Miranda House this opportunity to collaborate for this faculty development program. As has been said by our principal, this is the first collaboration with Ramanujan College, but we look forward to further collaboration. I would also like to thank Principal Dr. Vijay Lakshmi Nanda and Vice Principal Dr. P.K. Mishra, Vice Principal, Ramanujan College, for being with us and sharing their inspiring thoughts. This faculty development program has been made possible, apart from our collaborators, it is there and existing and running because of our resource personnel who have taken time out of their very busy academic schedules to share with us video recordings of their lectures. Our speakers are, through the 2 weeks, Professor A.K. Bhagi, Dr. Anant Narayan Bhatt, Professor Ashok K. Ganguly, Professor Ashok Kumar Prasad, Dr. B.S. Balaji, Professor D.S. Rawat, Professor Ganapati Subray Shankarling, Dr. Himanshu Ojha, Professor Jitendra M. Khurana, Professor Mangla Sundar Krishnan, Professor R. Nagarajan, Professor Rajiv Gupta, Professor Rita Kakkar, Professor R.K. Sharma, Professor Ruchi Anand, and Professor Vibha Tandon. Our eminent scientists and resource personnel, academicians are from across India, from very, very distinguished institutes, the Indian Institutes of Technology, Delhi, Bombay, Madras, University of Delhi, Jawaharlal Nehru University, New Delhi, the Defense Research and Development Organization, the University Department of Chemical Technology, Mumbai, to name a few. Some of these lectures, including the keynote address after this inaugural session, will actually be on the virtual platform. Our special thanks to the technical staff of Ramanujan College Teaching Learning Center for their invaluable support. I thank the over 400 participants, all faculty in colleges, universities, academic institutions across the country who have registered for this course. We are sure that this course is going to be an extremely inspiring and enriching experience for you. Before I end, just a few important announcements which are part of all faculty development programs. The first announcement, attempting and submitting all the quizzes and assignments is mandatory. Each participant should score a minimum of 50% in total to enable her or him to get the certificate of completion. Graded certificates will be awarded to all the participants. The grade depends upon performance. As part of the Ministry of Education's requirements under this scheme, the Pandit Madan Mohan Malviya National Mission for Teachers and Training Scheme, participants must submit the online feedback form for each session. Participants unable to meet the above requirements may miss out on getting their completion certificate. Though we are sure that that will not happen, but since it is mandatory for me to make this announcement, I'm doing so. This brings an end to our inaugural session. We will now have the keynote address by Dr. Anant Narayan Bhatt, who will be, as you will realize, speaking on a very, very contextual current topic, the base of which is, of course, chemistry. I hand over this virtual platform to my colleague and teacher in charge, Chemistry Department, Miranda House, Dr. Sharda Sonkar, who will introduce the session and, of course, our very eminent scientist. Best wishes to everyone and namaskar.
Thank you, Amrita ma'am. I would now like to introduce Dr. Anant Narayan Bhatt. So on behalf of the organizing committee, I welcome our keynote speaker, Dr. Anant Narayan Bhatt. Dr. Bhatt is working as Scientist F at the Institute of Nuclear Medicine and Allied Sciences, DRDO, Delhi. Dr. Bhatt has done his PhD in biochemistry from the Molecular and Structural Biology Division, Central Drug Research Institute, Lucknow. Dr. Bhatt has been a visiting scientist at Systems and Cell Biology of Neurodegeneration, Division of Psychiatry Research, University of Zurich, Switzerland. He has been a visiting Fulbright Scholar at the Department of Environmental Health Science, Bloomberg School of Public Health, Johns Hopkins University, USA. His research interests include the understanding of the mechanism of metabolic and inflammatory regulation of cellular stress response, mainly pathogenic, and radiation-induced stress and development of hemostatic formulation to combat casualty. Dr. Bhatt has published more than 40 research papers in various peer-reviewed journals of international repute and has contributed 10 chapters in different books. He has delivered more than 50 invited lectures at various conferences and webinars. Dr. Bhatt is also a member of the Indian Biophysical Society and the Indian Society of Biological Chemists. Dr. Bhatt is the recipient of many prestigious awards: European Molecular Biology Organization Fellowship Award from European Organization in 2012, Fulbright Nehru Senior Research Fellowship Award from the United States India Education Foundation in 2013, Laboratory Group Technology Award in 2019, Technology Day Oration Award 2019, and Science Day Oration Award 2020, to name a few. Participants are requested to kindly type their questions for Dr. Bhatt in the chat box. I now invite you, sir, to deliver the lecture on 2DG, an anti-COVID drug, journey of an age-old molecule to a medicine for the current pandemic. Welcome once again, sir. The Zoom is now yours.
Thank you, ma'am. Thank you, everybody. And thanks to the principal Miranda House and the principal Ramanujam College and Delhi University to provide me this platform. See, whenever we go through university, I mean, this is a virtual platform, but whenever we go through university, we always feel that the feeling of the student of the university comes. So except very few people who are fortunate and who get the opportunity to teach at the university, rest all the population of the country always feel whenever they go through university that they are the student. So I'm also one of that person who whenever go through university feel like a student. So just like as student only, I just want to share some of my scientific experiences and the development of this drug, which our team has developed in DRDO. So I would just like to share my screen. Is it visible to everyone?
Yes, sir.
Yes, it is visible.
Yes, sir. Yes, sir.
And it's in a presentation mode?
Yes, sir.
Okay, so we are going to talk today about 2DG as an anti-COVID-19 therapeutic from an old age molecule to a modern antiviral therapy. When I say it's an age-old molecule, then I mean, we discussed a lot about the chemistry, and I come basically from a biochemistry background. So there was a lot of discussion that the chemistry and its application in drug development, in biology, in life sciences. So I come from that hybrid background, and basically we use the power of chemistry to develop the applications in biology, and that's what this talk is all about. So before going to the development of this molecule, I just want to talk briefly about what basically triggers us, what basically the contribution of science which have happened till now, and which plays an important role in development of this application. This is a very well known, I mean, those who have read elementary physics, everybody knows about Kirchhoff's law. So this Kirchhoff's law tells about the current coming in any circuit, the sum of the current which is coming in any circuit, and the sum of outgoing current is always equals to each other. But somebody on social media posted this photograph and asked one of my friend and asked, can you apply Kirchhoff's law to this circuit? So this was a very difficult circuit. I mean, but still you can see that that how many wires are there. Maybe visibly it looks very difficult to solve this puzzle, that what is the incident current and what is the outgoing current, and solving it becomes very difficult. But still you can see all the wires. Suppose that somebody spent maybe whole day or few days, then probably able to solve that that what is the current which is coming in and what is the current which is going out. So but one of those circuits which are very complex than this. So probably this circuit, which is called cellular biochemical pathways, this is even complexer than this. The complexity of this is even higher grade, because you can't see anything. You have to conduct experiments, and those experimental evidences, which are indirect evidences, based on that you have to conduct several experiments to conclude anything, that probably this molecule, this chemical molecule, this biochemical molecule is getting converted to this biochemical molecule, and based on which a lot of biochemists have put their entire life to I mean, conclude that probably whatever understanding we have till date, that probably this is the cellular biochemical pathway. I mean, these things are still evolving. This is not the final one. These things are still evolving. And my talk, today's talk, is tribute to all those biochemists who dare to solve this probably most complex circuit which is evolved in million years of the evolution on the earth, in the form of human cell or mammalian cell, you can say. So one of those scientists, and he was a physician, actually a German physician, who talked about that cancer. He basically worked on he was an oncologist, and he discovered a phenomena which is known as a Warburg phenomena in his name. And it says cancer, above all other disease, has countless secondary causes, but even for cancer there is only one prime cause, and that prime cause is enhanced glucose metabolism in cell. So the I mean, the utilization, the uptake of glucose in these cancer cells goes very high, and that is what is basically required for very fast proliferation of cancer cells. So the replacement of the respiration of oxygen in normal body cells, which takes place in mitochondria, gets replaced with fermentation of sugar. That time it was called fermentation of sugar. Later it was realized that even in the presence of oxygen in cancer cells, they keep metabolizing very high amount of glucose, which is generally called anaerobic respiration, and that happens generally in the absence of oxygen. So this, this was a very long puzzle for the scientists, that why this is not a very efficient pathway, not a very economic pathway to produce energy. Why the cancer cells are using this pathway, a glycolytic pathway, and not using mitochondrial pathway to provide energy. Later, lot of evolution happened. Warburg got a Nobel Prize for this discovery, and another scientist, Louis Sokolov, who also worked on the same phenomena and designed a PET imaging for cancer. You all must be knowing, like for advanced cancer, a lot of patients, they undergo PET imaging. In Mass is also one of the institute where PET imaging is being done. So Louis Sokolov also discovered a molecule that's called FDG, fluorodeoxydiglucose. And like all the cancer cells, they accumulate a lot of glucose. So fluoro is basically radioactive glucose. So it gets accumulated into the cancer. So when it is given and the person is taken into PET scanner, then where are all these tumors are situated. So that was a very remarkable discovery. And based on that, Louis Sokolov also got Nobel Prize, in 60s or 70s. And Warburg got very early, in 1930s. So this discovery was very old. And that is why I call that this molecule is an age-old molecule. We have just used it for a new purpose, that is a new application. So why glycolysis? Like whenever, as I said, this question comes to many people's mind, that this is not a very efficient pathway. It just gives 2 molecules of ATP if you will be using glucose into the cell. But what happens that the production of ATP through mitochondria is very slow. And suppose that if in 1 minute, 1 molecule of glucose is being metabolized through mitochondria and producing 32 molecule of ATP, 32 or 36 depending on different shuttles. So, but in the same amount of time, 1 minute time, if glycolysis is upregulated, it can convert 50 molecule of glucose into pyruvate or lactate, whatever, depending on the availability of oxygen. So what will happen from that 50 molecule of glucose conversion, it will produce 100 ATP. So of course it is not very economic, but it is very efficient. The rate of ATP production is very high. And that is why, like whenever then any emergency come, you all must be knowing adrenaline is known as a triple F hormone: fight, flight, and fright. So whenever we want to run, whenever we want to fight, whenever we are afraid of anything, that is the time when a body needs maximum amount of energy, and body switches to glycolysis, does not rely more on mitochondrial respiration. So this is what the evolution, this is what happened evolutionarily, that whenever you need large amount of ATP in a very short span of time, you need your body switches to glycolysis. And this cancer uses this mechanism, and then it converts the glycolysis to a very fast upregulate glycolysis and then converts the energy. I will give one more example. Like the fetus, no tumor can grow as fast as fetus grows. Like from a single cell in 9 months, we get a baby of 2.5 kg. No tumor can proliferate, no tissue, sorry, for using the word tumor, no tissue can grow at this pace than what a fetus grows. And in a fetus also, the glycolysis is very high, and that is how but it is regulated. The problem in cancer, the glycolysis is very high, but it is not regulated. However, in fetus, the glycolysis is highly regulated. But what happens even before birth, epigenetically, this glycolysis goes down. When the maximum growth is achieved, the epigenetic regulation happens, and then this glycolysis goes down in fetus, and the children who is born, the baby who's born grows on its normal pace. So that is what I want to bring the point here, that for fast growth, for fast biomolecular packaging, for making newer cells, you need lot of, you can see here, you need lot of RNA and DNA through triple P pathway. Whenever glucose comes in glycolysis, it goes, and you need lot of RNA and DNA is required for making the genetic material of the cell. And then you need lot of lipid, which through different pathways it comes, and then it forms lot of lipid, single carbon, through single carbon metabolism again, which is the source is glucose. So these things are required, and you must be knowing like these things. From single carbon metabolism, glucose is the source, and all these intermediates which are formed in glycolysis, they, after giving ATP, they further participate in several other biochemical pathways, and they contribute in anabolism. So, so what is the basic difference between glucose and 2-DG? So this is the molecule of choice today. You can see in the glucose at position 2, the only change which happens is there is no oxygen here in 2-DG. So what happens, like this molecule gets easily incorporated into the cell, uptake happens into the cell very easily, because it is almost like glucose. But what happens, like the first step of glycolysis takes place properly, but the second step doesn't take place, because at the second step, the conversion of glucose 6-phosphate takes place into fructose 6-phosphate, and fructose is a keto sugar. So oxygen is mandatory at position 2. So oxygen, if there will be no oxygen, this cannot be converted into from glucose 6-phosphate to fructose 6-phosphate. So this becomes an inhibitor of the next enzyme, and then what happens, like the glycolysis gets stopped there. So the cell, what happens to the cell? Cell think that ATP is not coming, so it will produce more and more GLUT receptors for more and more uptake of glucose. And what will happen eventually, because of that reason, it will accumulate more drug. So there will be selective accumulation of the drug, which will take place in this case. So this drug will accumulate more, and it will choke the glucose metabolism completely in those cells. And these kind of cells, like cancer cells and the virally infected cells, we'll be discussing in detail more about it. These kind of cells, they are basically, they specifically rely more on glycolysis, as I said. So their mitochondrial respiration will be blocked. So their dependence on glycolysis will be very high. And because of that reason, once glycolysis will stop there, what will happen that this energy production and overall metabolism in terms of catabolism and anabolism both will stop, and that is why it will not be able to, those cells will not be able to do their proper function, that is like synthesis of new cell or synthesis of new virions or new virus. So no oxygen is there at position 2. It mimics glucose in plasma and cellular uptake. So all those people who have worry that what will happen, this is a drug, whenever you take a drug, you worry a lot about that what will happen to normal tissue toxicity. But as I said, it mimics exactly like glucose. So there is no concern outside the cell. So there is no concern of side effects or tissue toxicity. But the only thing that it does not metabolize when it gets internalized into the cell, which is I'm showing in the lower portion of the of this slide. And it inhibits anabolic reprogramming of the cell, so that more and more synthesis of biomicromolecules cannot take place, and virus synthesis gets affected. So this is what about the cancer, like this is the background and the journey of this molecule, which took place in mass since decades. And you can see that normal cell division takes place at normal pace, and it is a damaged cell, one cell that gets damaged, and then it becomes cancerous. Some of them, if they are not dead, they will become cancerous. So this cancerous cell will have then very fast proliferation, and with high proliferation, their glucose demand will also go very high. And this proliferation can be stopped by using 2DG. So 2DG, what it basically does, it make the cell, cancerous cell, cytostatic. It will stop the proliferation and the cell will not be proliferating. So this was the, but it will stop there. If you want to kill the cancer cells, if you want to further reduce the tumor volume, then you have to apply radiotherapy or chemotherapy along with 2DG. And that's what INMAS has been working on this and have done since ages. Dr. Dwarkanath and Dr. Vinay Jain, they have long collaboration with Delhi University. I think the old faculties must be knowing these two scientists who worked for a long time at INMAS. Dr. Jain was former director of INMAS, and Dr. Dwarkanath was former additional director, and both are retired now. And they brought this molecule to, Dr. Jain brought this molecule to India, and they worked for a very long time on this molecule to develop the application in cancer. So they conducted phase 1 and phase 2 clinical trial at very reputed institutes like AIIMS Delhi, TMH Mumbai, Kidwai Memorial Hospital Bangalore, Nizam's Institute of Medical Sciences, and Dhamshila Cancer Hospital Noida. The trials were conducted on these medical centers. What happened, like in 2004, due to some reasons, we joined hand with we had to synthesize the 2DG in India itself. And that is what the current technology DRDO now has. Because what happened, like in 1998, when Pokhran blast happened, that was the time when DRDO was buying 2DG on a bulk level to conduct the clinical trials. So US put sanctions on all those things which India was buying that time from US. So 2DG was also in that list. So we could not buy 2DG from USA. So one of our chemistry lab that is situated in DRD, in Gwalior, it's called Defense Research and Development Establishment, DRDE. So that is specialized in chemistry, and they have synthesized 2DG for us. And based on this indigenized 2DG synthesis through a new pathway, new chemical pathway, we transferred that technology to Dr. Reddy's lab in 2004. And then we conducted the phase 3 clinical trial in cancer patients along with Dr. Reddy's lab. Then in later 2014, DCGI recommended that this drug can be used for glioblastoma multiforme patients. This 2DG drug. So now coming to the virus. So what is the, so this was the background. I mean, our previous work on 2DG in cancer. Recently, when in 2020, March, when this problem, when, his cases started coming in India, there was a lot of brainstorming was going on that what can be done. So the advantage with us was like, if this molecule can be useful to inhibit the virus proliferation, SARS-CoV-2 proliferation. And the good thing with us was that we have already tested this molecule in clinics in cancer patients. So that experience was with us. And we thought that probably that experience can help us in translating this molecule very fast to fight this pandemic. So, what is the rationale behind that? Is this, like in virus, there is nucleic acid. So, by inhibiting glycolysis, you can inhibit the nucleic acid synthesis. So virus cannot proliferate very fast. Then for lipid synthesis also, it has a lipid wall. And that is why everybody is asked to wash the hands using soap, because soap will, solubilize these lipids and the virus will die. So the lipid synthesis, lipid is also one of the constituent molecule of SARS-CoV-2. So it cannot, if you can inhibit this lipid synthesis, then also you can target this, you can target its replication. then it is made up of phosphoprotein, which also requires ATP. So by inhibiting glycolysis, you are compromising ATP synthesis as well. So what will happen? There will be no ATP and there will be no phosphorylation of these proteins. So what will happen that the overall replication or multiplication of SARS-CoV-2 will be affected. So this is what I have already explained. So while oxidative phosphorylation provides significantly more ATP per glucose, glycolysis is a much faster way of providing ATP, and that is why evolutionarily all these cells, even virally infected cells, also uses glycolysis, high glycolysis, for more and more virus production, virion production. So the basis of using it is that upon infection, virus hijacks and reprograms the host cell metabolism. So, you know, like all our normal cells, they need very less amount of glucose, and they produce more amount of ATP using oxidative phosphorylation in mitochondria. But whenever virus infect the cell, what happens? That mitochondrial metabolism is completely shut down because of the damages which are caused because of viral infection, and that damage comes from dysregulated calcium homeostasis in the cell. So because of that dysregulated calcium homeostasis in the cell, mitochondria gets affected. So mitochondrial metabolism also gets completely affected, and in response glycolysis goes high, and that's how, which is required for more and more viral synthesis. So unlike normal cells, which show more plasticity in their metabolism by relying on both glycolysis and mitochondrial pathways, SARS-CoV-2 infected cells operate predominantly only on glycolysis, or you can say more than 90% on glycolysis, because for cell survival, mitochondria is also essentially required. So those cells, to survive, they need mitochondria, and that's how bare minimal function or biochemical pathways in those virally infected cells goes on. So the anamolic demand is predominantly being fulfilled of virally infected cells, is being predominantly fulfilled by glycolysis predominantly, and that is why normal cell will not be much affected, because in normal cells, even if you inhibit the glycolysis, those cells will survive on mitochondrial metabolism. But the mitochondrial metabolism is not there in virally infected cells, or very minimal is there in virally infected cells. So if you inhibit the glycolysis, they will be significantly compromised for replication of virus. So this is a presentation, presentation where you can see that in normal cells, what is happening, basically glucose is coming and then it is contributing to nucleotide biosynthesis and then ATP production, then it goes to the mitochondria and lipogenesis and other functions takes place normally. But in a virally infected cells, when infection happens, you know, glycolysis goes very high. As I said, you can see like all these receptors, GLUT1 and GLUT4, and recently we found out GLUT3 also, which goes very high. So GLUTs are basically transporters of glucose to these cells. So, you know, like those who have studied biochemistry, they know that all these transporters, enzymes, they have a Km value. So Km value determines at very low concentration, how low concentration some enzyme can bind with the substrate and can perform the reaction. So these GLUT1, 3 and 4, their Km values is very low for glucose. So even if a small molecule of glucose or 2-DG is available, they will immediately pick up that and they will accumulate that into the virally infected cells. So because their expression goes very high. So you can see here that the level of these transporters goes very high, and because of the increased level of transporters, the large accumulation of this drug happens. So the one thing is, what is happening is the selective higher accumulation of 2-DG is happening in virally infected cells. So this mechanism is a very beautiful mechanism that you don't have to make any change to target the infected cells or to target the tissue of interest or cell of interest. So this molecule inherent has this targeting that it will selectively accumulate, high accumulation will take place in virally infected cells. That is one thing. And then 2DG inhibits energy production. So once this will be taken up, it will inhibit energy production. We discussed in detail about it. And then 2DG inhibits anabolic biochemical reactions like the synthesis of nucleotide and lipid and so on. So due to which the more viral replication cannot take place. And then another mechanism which we found out, which was reported in literature in case of cancer, but in case of virology, we have found out that 2DG alters glycosylation. You know, all these secretory proteins. So a virus is also a secretory organism. So it has a lot of glycosylated proteins. So those proteins, the sugar moieties are added on their proteins. They're, you know, like all these blood groups. So O, A, and B is what basically they are different glycoproteins, and the glycosylation is different in A, B, and O, and that is how they become, these blood groups are different to each other. So similarly, these viruses are also having glycoproteins. So 2DG, I mean, you know, like mannose is epimer of glucose. So the only thing is, like in glucose, at position 2, OH is below and H is on the other side. I mean, what I'm telling, looking at I mean the picture or structure of this. So in mannose, what happens? The H is just like epimer, so H is below and OH is on top. So what happens, like in 2-deoxyD-glucose, I mean OH is not there, so it is only H. So it mimics the mannose also. So what happens, the cell takes 2DG and it incorporates, it gets fooled and it incorporates as a mannose. So because of that, misglycosylation happens. And that misglycosylation is something like you are writing address on a letter, but you misspell the address, or you write a wrong address. So the letter will not reach to the right place. So because of that reason, what will happen, these viruses which are produced, so these glycoproteins are kind of an address which is taking this virus to ACE2 receptor. You all must have heard a lot about that. These viruses, they go and bind to the ACE2 receptor of our cells, which is and the cells of the lungs, cells of the esophagus, and cells of the heart, kidney. Some of them, they have very high level of such cells, which is expressing high level of ACE2 receptor. So because of that, these organs are very susceptible to SARS-CoV-2 virus. So this address is basically directing it to ACE2 receptor. So because of misglycosylation, what will happen, that this virus will not be able to reach to the right place, or even if it will reach, it will not bind to ACE2 and it will not be able to infect the new cells. So whatever, like first thing this molecule is doing, it is inhibiting the replication of the virus, let's say 95% at any given concentration. So whatever remaining 5% viruses are being formed, they are not able to reinfect the new cell. So that is a very, this is the second mechanism, and due to which we are getting a very good response. So this is the experimental validation of whatever we have talked. So 2DG selectively accumulate in virally infected host cell. So you can see like this is a ciliated lung cells, and the first cell and the fifth cell got infected with the virus. Not all the cells gets infected with the virus. So first and fifth cell get infected with the virus. And as I said, like because of, so this is a model, simple model to explain. So 2DG accumulation will take place maximum. This green color is, green dots are basically 2DG. So they accumulated maximum into the virally infected cells, but not in normal cells. So that is how they are affecting, only selective accumulation is happening. And you can see here that as compared to uninfected cells, infected cells accumulated much more amount of 2DG. So this green color is basically coming from a fluorescent analog of 2DG, which is called 2NBDG. So N is basically, I mean, it's a compound which is tagged at position 2. It is again the analog of 2DG, and it is a fluorescent analog of 2DG. So when you stain the cells, so here it is a co-culture. These red color cells are those cells which doesn't take viral infection. And the green color cells which you are showing in the right side are all those cells which takes viral infection. So so this is a co-culture. That means in the same culture we have cultured both kind of cells, which is mimicking the actual situation in our normal tissue, where only very few cells will be infected, not all the cells. So when we mimicked that kind of situation, you can see that all those cells which are not showing red, that means they can take viral infection, they all have very high accumulation of green color, which is coming from 2-NBDG, which is the fluorescent analog of 2DG. So that means that these infected cells, they take up lot of glucose. The accumulation happens selectively very high in only those cells which gets infected. So this resolves the concern of side effects of this drug. So this is the very first evidence about the safety of the molecule. Then about the efficacy of the molecule. So this experiment, like in the beginning of beginning of pandemic, we proposed that this molecule can be very effective, and probably we can develop a drug because we have experience of clinical trial with this molecule. So I got the opportunity to present this proposal in front of Dr. V.K. Paul, who is heading this entire program of COVID management from Niti Aayog. So I presented in front of him and he approved this proposal. And at that time only CCMB Hyderabad and NIV Pune were conducting, were only had this virus to test any drug. So they have established this test. So I went to CCMB Hyderabad and we conducted this experiment, and we found that this 2DG is very significantly able to inhibit the viral replication, and this is in supernatant and this is in cell bound. So at very low concentration, which we have used for in cancer, much lesser concentration than that, we found that it is able to inhibit the viral replication. So the second hypothesis, this is again an in vitro experiment. So second hypothesis, like the whatever virion which will be produced, that will not have, what I was talking about, the misglycosylation, and that will not reach to the right place, and it will not be able to infect the neighboring cells. Because this is what happens, like when cells gets infected, those cells, they die and they release all the virions. Even when they are surviving, they will be keep releasing the virions, and these new virions, they will go and infect the neighboring cell, and that is how the whole lung or the infection is spread in the body completely. So once this drug will go to selectively go to infected cells and inhibit the viral replication, and then whatever released virions will be, they will have misglycosylation, and they will not be able to infect the neighboring cells. So this experiment we performed, like we treated the cells with 2DG, and then reduced concentration of virus was obtained, and then we normalized the concentration of those viruses and reinfected the cells. So when we reinfected the cells, we with the progeny virions, you can see that the progeny virions were not able to produce the same number of viruses. So there was 80% reduction in the on the same concentration of viral infection. So that means these they have the progeny virions of 2DG infected cells, they lost the infectivity, they lost the capacity of infection, and that is why that this drug is very effective in controlling the viral infection. See, the other concern lot of people had, that because 2DG is used to kill the cancer cells, so probably this 2DG will kill the virally infected cells as well. So but as I said, like it can only make the cell cytostatic. That means cells will not proliferate, but for killing the cell, you need additional thing, that is radiation or chemotherapy agent. So that is very well established in cancer biology. So we conducted those experiments again, and you can see here that in the middle row, which is infection, all these red colors are suggesting lot of cell death. There are a lot of dying cells can be seen here. But in the top row, which is normal cells, very few cells minimal cells are, this is control non-infected, very few minimal cells are shown as a dead. And as compared to infection, when 2DG is given, you can see like because 2DG is able to inhibit the viral replication, the cell death is also significantly reduced in this. So it is not additionally causing any cell death from coming because of 2DG. And you know, this inflammation, you must have heard that lot of debate on that the inflammation goes very high in COVID patients, and that hyperinflammation is causing death in many of the patients. And you know, like all this hyperinflammation also comes from the very rapid high number of cells dying in the lungs. So lung is very sensitive to inflammation. So I mean, if lot of lung cells will die, the inflammation will become hyperinflammation. So 2DG is not able to only reduce viral replication. By reducing the dying cell, it will be able to control the inflammation as well. So so this is the observation we got, and this is the summary of this and all the experiments which we have performed, in vitro experiments. You can see in the top one what is happening. SARS-CoV-2 infection is happening, and then normally it is taking replication is taking place, and then it is being released from those infected cells, and I mean affecting those neighboring cells, and all the cells are dying, and because of that inflammation is happening, and the worsening, the pathology of this entire COVID is happening. However, in the lower panel you can see that with infection we treated the cells with 2DG, and it stops the energy production and anabolic reactions, and then it causes misglycosylation. So because of these reasons, the cells are not, I mean the viruses are not able to replicate, and whatever small amount of replication is happening, it is forming a weak virion, or I mean misglycosylated defected virions, and those defected virions doesn't have capability of infecting neighboring cells, and that is why the cell death is not happening and viral replication is stopped there itself. This is the data not from our lab, from some other lab, where people have shown that 2DG is very efficiently able to inhibit the inflammation, lung inflammation predominantly. And this lung inflammation is not caused by virus, it is caused by LPS, which is a bacterial product. And you can see here that IL-6, you all have heard that IL-6 is one of the marker of COVID cases also. The IL-6 level goes very high, and that is causing hyperinflammation and cytokine storm, and people are dying because of that. You can see here that plasma IL-6 level also goes significantly down when 2DG is used when given to the mice. And the pulmonary, I mean, this is pulmonary IL-6 level are basically measured by taking out the BAL fluid, bronchoalveolar lavage. You take out from the lung and then you measure the IL-6 in that, and that level also has gone significantly down. And you can see the mortality rate on the right chart, survival Kaplan-Meier plot. You can see that the mortality has also gone significantly down when 2DG is given very early. So this suggests that it is not only affecting the, the viral multiplication, it is controlling the inflammation also, and that is why the drug is very effective. This evidence is published in PNAs in 2018, where this information is coming from rhinovirus only. This is another experiment in animals, in mice, where people have, infected. The rhinovirus is again causing the respiratory infection. And you can see that all inflammatory markers has gone significantly down in presence of 2DG. And this is on the right side, you can see that this is the lung morphology and histochemistry of the lung. And you can see here that the cells are, completely, this is H&E staining, and the cells are, the lung is completely damaged and a lot of fibrosis is taking place. Whereas when it is treated with 2DG, this fibrosis is reversed, and then the cells, the inflammation is not there in the lungs. So this is a very, this is a very good evidence which suggests that it can control the inflammation as well. Then another problem which is happening with COVID is the infection. Recently you would have heard about the black fungus and all. And we don't have any laboratory evidence. We are working on it on those in those lines. But there is a lot of literature evidence which suggests that this can control even, I mean, because, you know, like for any proliferation, even for fungus proliferation also, it need biomass, it need biomolecules, and 2DG can inhibit that also. And it is shown that it can inhibit the growth of yeast, which is again a fungus. And we found that the yeast inhibition was seen in our clinical trial data also. You can see like the patients who were recruited with yeast cells in their urine infection they had, those patients, when they were discharged, they didn't had yeast infection. Whereas the patients who were treated on standard of care, the standard of care basically means all those drugs like remdesivir, favipiravir, ivermectin, etc. They when they were discharged, they did not had viral infection, but they still had these yeast infection. So that's how this drug is very potential to even working on even on the fungal infection as well. So with all these data, I mean, we didn't have these much data when we got the approval for conducting clinical trial, but I mean, eventually we collected all these data for mechanistic study, and then in the May itself, we based on the CCMB, the experiment we conducted in CCMB, DCGI has Drug Controller General of India has given us the permission to conduct phase 2 clinical trial, which we conducted in 110 patients in 2 different phases, sub-phases. And based on the very good efficacy results, we got the permission to conduct phase 3 clinical trial in November 2020. And we conducted that study, which has gone up to April. And in April, we got, I mean, some preliminary data, and we analyzed those interim preliminary data. And then we presented that to DCGI. And DCGI has given us the emergency use approval to use this drug. So in phase 2 and phase 3, what we did, like we had 2 group of patients. In one group, we have given 2DG, 90 mg per kg per day, and with the standard of care drugs. And in other arm, we have given only standard of care, which includes all the antivirals, which I told you, which I mentioned. And the sample size were 220 patients. And based on these results, what we got, like there was 2 to 4 days reduction has been observed in 90 mg, in the drug 2DG, in hospitalization, in oxygen saturation is also very early, very fast. And, you know, that was the major problem. Like the longer you keep the patients on oxygen, the survivability or chance of the patient coming back is very minimal. Because, you know, like high pressure oxygen is not good for lungs, for any cell. So just to fight with the disease and that kind of pathological conditions, we have to use that high pressure and high concentration of oxygen. So the faster we bring the patients from the oxygen support, the better the patient will recover from the disease, that is COVID-19. So this showed very nice change that at day 3, almost 50% patient came out of oxygen supplement as compared to 30% patients in standard of care arm. So there was a big statistical 20% difference was there. So 2DG was approved for emergency use as an adjunct therapy. And it is prescribed mainly by the doctors to moderate to severe patients for maximum duration up to 10 days. And because uncontrolled diabetic patients and patients with severe cardiac problem, ARDS, they were not recruited to the clinical trial. So currently, after complete approval will come, then only probably we can permit that these patients can be given with caution. So 2DG should not be given to pregnant and lactating women because they were not the part of trial. So we will, they should also be avoided. And this comes in this small sachet. You can see like this long work of several decades has finally come up in this small sachet. And this sachet can be dissolved in 100 ml of water, and it can be taken morning and evening. 2 doses can be taken. and per day, and the maximum treatment goes on up to 10 days. But for many moderate patients, we have seen like those patients, they were discharged by day 5, day 6, day 7, like that. So the treatment can go maximum up to 5 to 7 days. And a lot of people, I mean, more than 50% patients were turned negative on RT-PCR on day 3 itself, between day 3 and day 4. So so that is another good advantage. So moderate patients probably don't need more than 5 to 7 days, but some critical patients may need the drug up to 10 days. And this is this whole work, this entire work could be successful, especially this anti-COVID program, which we led by our chief, chairman DRDO, Dr. G. Satish Reddy. I'm grateful to him. He always constantly supported from all the angle, you know, like when it was difficult to come out of the house during the first wave in lockdown. Our labs were fully running, and we conducted these experiments. We performed a lot of experiments, and I was sent to CCMB Hyderabad by a special flight, so to conduct the experiment in virus at CCMB Hyderabad. So all these facilities and all this support came from our chairman, and his vision and leadership to fight this pandemic. This is the national, emergency situation. And so a lot of credit goes to him. Then Dr. Jain and Dr. Dwarkanath, who worked a lot on this molecule. And if that clinical evidences which they have worked on cancer would not have been there, probably we could not have been translated this molecule so fast in just 1 year. I mean, clinical trial is not a job which can be done very fast in 1 year. And you know, like all these molecules which came to the market, to the clinic, like remdesivir, favipiravir, they were already in the market. This is the only drug 2DG which was not in the market, which has come till now. So 2DG was not in the market. It was a lab molecule, and it was being done. But of course, a lot of clinical trial were done on this, but it was not available in the market. So we brought this molecule with a very fast pace. It's all because of the clinical trial experience which come from Dr. Jain and Dr. Dwarkanath. Dr. Chandna, who is my head, and he helped and supported this program very well, and he is one of our team member, senior person leading this program. Then Abhishek, Yogesh, and Neeraj, these people, they have did lot of experiments. As I was telling you, like when during the lockdown, these people were continuously coming to the lab, and initially when a lot of people had fear of working on the virus, we got training in NCDC, and then we started working on this molecule. Handling the SARS-CoV-2 virus. So there are a lot of other people also in INMAS and DRD, Gwalior, who synthesized that 2DG molecule indigenously, and DRDO team, and Dr. Reddy's laboratory also. They also work day and night along with us to conduct the clinical trial and to realize this molecule which could come, which we could brought to the clinic. Thank you very much. And I'll be happy to take any question if it is there.
Thank you so much, sir, for this enlightening lecture. I'm sure we all can feel much safer with this 2DG now. I would like to now invite Dr. Mamta Sethi to kindly anchor the questions from the participants.
Thank you, Shada ma'am. Thank you, Dr. Bhatt, for sparing your valuable time for this faculty development program. And I'm glad to share that the chat box is overflowing with the compliments that clearly shows the gravity with which you kept our participants engaged, sir. We have a few questions in the chat box. With your permission, I'm asking you on the behalf of audience, sir.
Definitely, ma'am. Yeah.
So there is a question from Mr. Sonu Ram Shankar. It appears to have worked through a common mechanism.
can these results be extended to any other viral infection? If not, why?
This is a very good question. As I said, like, it is not working on any virus specifically. It is working on host cell metabolism. So not all the viruses, like I mean, people have shown that this is a well-established phenomena that glycolysis goes high when viral infection takes place. But it doesn't happen with all the viruses. And you know why defense is more interested in this program? Because, you know, like when it came in December, it took, I mean, the first drug, remdesivir, came somewhat around July. So it took almost 6 months, 6 months to 7 months time to bring any molecule. So if this drug is approved, it will be a non-specific molecule, and it can be given to vast majority of antiviral viral infections. So of course, this molecule will be a non-specific antiviral molecule, and it probably will be able to work in other viral infections as well. And that is why defense is more interested in this, to use it as a tool for to fight against bioterrorism, where the virus will not be identified. I mean, we'll be able to come to know about the virus much later.
Thank you, sir. I hope the participants are satisfied with the answers. There's another question from Mr. Parveen Singh. Does 2G come from outside the cell, and what is the end product in the human body?
See, its metabolism is not very well understood, but of course it comes from outside. I mean, when we give it orally, it gets absorbed like the absorption of glucose, and it gets absorbed into the blood. And its pharmacokinetics is like this, that up to 60 minutes it gets peak, and then it comes down. And that is the pharmacokinetics and blood clearance, and it gets accumulated to all those cells which is demanding high glucose. So it will get accumulated, and it will get metabolized to 2-deoxy glucose 6-phosphate. So it gets accumulated predominantly as a 2-D glucose 6-phosphate, and it remains there for a very long time. But its further metabolism or degradation is not yet studied.
Okay, thank you, sir. There's another question from Dr. Pratap. Is the action of 2G related with the already existing vaccines, Covaxin and Covishield?
I mean, in our trial, a lot of patients were recruited, those who were already given vaccine. So, I mean, it doesn't have any interaction with it. You know, like, it is a glucose. So it is just like glucose. It having only, only one oxygen, which is less. So, so its interaction with any other things is not known, and it can be given with the people who have taken vaccine.
Thank you, sir.
If I corrected the question correctly, I think, I don't know if I understood the question correctly.
No, sir, we understood, we got it. And there is another question which comes from Dr. Anbu Srinivasan. He needs a clarification regarding the side effects of 2G when it is taken in initial stages of COVID-19.
Yeah, so the only one thing which we have seen, like around 3 to 7% patients we have seen in different studies, that when taken, they develop little perspiration, I mean, sweating and little uneasiness. That happens after 1 hour, 45 minutes to 1 hour after taking the drug. And it remains there for, let's say you can see, for 15 to 20 minutes. It happens after 1 hour, and it remains there for 15 to 20 minutes. And if and what happens, like, immediately after taking this, after 1 hour, 1.5 hours, there are a lot of people who feel very hungry, so they start eating a lot. So these are the things which we have observed. but otherwise no, I mean, no significant side effects, SA, which we called AEs, adverse events, was observed in all clinical trial.
Okay. So I think you mentioned about the pregnant women and lactating mothers. Some participants have asked this question, that why specifically this drug cannot be administered to pregnant women and lactating mothers.
See, as a, I mean, ethics, no trial should be conducted on pregnant women and lactating women, because I mean, you know, like kids are very susceptible to any new drug. So, I mean, when the safety of that drug is not established, so clinical trial is not conducted on these patients. And that is why, because clinical trial is not conducted, it is not permitted. But it completely depends on the wisdom of I mean, the treating physician, if they want to. And as I said, I give example also that fetus is one of the very fast proliferating tissue, right? So if you inhibit the glycolysis for very long time, see, if you are inhibiting for short time, like for just few days, then it is not a problem. But when the glycolysis is inhibited for a very long time in those fetus, then probably it is not good for the fetus growth. I mean, this is what we assume. We have not performed any study on this, but theoretically this is what we assume. So even if suppose that somebody want to use it in pregnant women's, it will be, I mean, the doctor should take proper caution when using. I mean, if they want to use it, they may use it in probably in the last trimester. That is the best way, but initially it should not be used for sure. Thank you,
sir. There are, of course, many questions.
And for lactating, I just want to add, and for lactating women's, it should not be a problem, because, you know, this is not a toxic drug. So even if it goes in the milk, it should not be a problem.
Sure. Thank you, sir. There are so many questions, but having time constraints, I would just like to take one more question. And then with your permission, if you can share your email ID, sir, we can share the questions from participants with you through email, or if you would like to share your email ID directly with the participants, they would like to ask questions themselves. So the last question which I take is that, is there any relationship with the efficacy of the 2DG drug and the blood group?
I mean, we have not performed any such a study, and I don't think it affects. Like you see, we have recruited 220 patients with a very vast different blood group. So, and we didn't found any difference. I mean, the average outcome of efficacy was more or less similar throughout the blood groups. So I don't think it is going to affect. Yeah. And my email ID is there with the organizers, and I can spell out also. It is anant@inmas.drdo.in.
Thank you so much, sir. Thank you so much, sir, for taking all the questions so patiently. And I mean, we are truly enriched with the knowledge as you've hit the right chord at the right time. This is something we all are worried about these days. Thank you so much.
Thank you very much again to the organizers for giving me this platform. Thank you very much.
Thank you, sir.
Thank you, sir. Ravinder. So thank you. Done for today.
Okay, thank you, Dr. Ashish. Thank you, ma'am.
Thank you. Thank you. I think even though
assignment. Anshika ma'am, please send the quiz and assignment.
Yeah.
quiz मैंने बना ली है. Sadha ma'am.
Yeah, Anshika, I think just give me 15 minutes. I'll send it to you.
So an assignment is ready now. Ashish, I've already sent you the last night.
Assignment, na, right? To send the assignment.
Assignment. Day one assignment I've already sent you, and quiz I'm going to send in a half an hour.
We can stop screening.
Ashish, still we are live.
Yeah, that's what I was about to say. We are still live.
If I can just ask, our next live session is on Sunday, isn't it?
Yes, ma'am.
So will we get another Zoom link, or is it the same Zoom link for Sunday?
Ma'am, no, we have another Zoom link I've posted in WhatsApp group.
Oh,
I've posted
sorry,
all the
Yeah,
Zoom
all
links
the
regarding-
Zoom session link, yes, ma'am.
sorry. Then I missed it. Thanks.
Dr. Ashish, we are still live on YouTube. Will you please ask admin to stop the live screening?